Alright, for movable substances, we're going to learn three very important processes. First one is diffusion, second is osmosis, and third one is active transport. So, like I mentioned, before we start, we will have to go through four very important factors first. These are used in the definitions of each of these terms, and it's easier to help you remember. So the very first kind that you actually see here is that there is a P. P is actually referring to particles, and these particles, type of particles, they could be water molecules or any other particle. Gradient, and the gradient type that you're looking at, whether is it the concentration gradient or is it the water potential gradient. Is there any energy required? And was there a partially permeable membrane involved as well? So for diffusion, what we are looking at is any particles except water molecules. The gradient we're talking about is actually a high concentration to low concentration. We're not talking about water molecules, and there's no energy involved, with or without a partially permeable membrane, describing it as a net random movement of the particles from a region of higher concentration to lower concentration down a gradient. What exactly is a gradient? Diffusion talks about concentration gradient. That means the concentration gradient we are looking at is of a certain particle X. You can see here is actually a high to low concentration. This is actually the gradient that we are talking about, like the gradient of a slope. And the steeper gradient here, as you can see, compared to the next one, which has a gentler concentration gradient. Right? So what if both of sides are the same, where you have equal concentration of the molecule or the particle X? There is no concentration in this case, hence there is no net diffusion. Alright, no net diffusion means overall gain or loss of particles, okay, is zero. So for diffusion, a few examples will be the gaseous exchange in lungs, at the leaves, and the ileum. Let's cover them briefly. Please do not memorize them, alright? If it's a new situation, you'll be given a story to read. Through a membrane gaseous exchange in the lung, we talk about how the oxygen is being taken in and how carbon dioxide is being removed from the lungs. Through the membrane gaseous exchange, the leaf is at the stomata, where we talk about diffusion of the carbon dioxide as well as the diffusion of the oxygen gas. Through a membrane oxygen at the ileum, which is the small intestine, we are talking about the absorption of the end products of digestion. It may seem fast that I'm talking about diffusion, but actually it's very straightforward. Okay. Diffusion, a few factors affecting it will probably be the first one, the temperature. The higher the temperature, the more kinetic energy the particles have, that means the ability to move. The faster they move, the faster the rate of diffusion. Smaller particles or smaller molecules tend to diffuse much faster, of course. Thickness of the barrier. The thicker the cell wall, the slower the rate of diffusion. The thinner the cell wall, basically, the diffusion also goes faster. Alright? Concentration gradient, of course, the steeper the gradient for a substance, the faster the rate of diffusion. It is the same thing as rolling down a very steep gradient from a slide compared to a very gentle slope slide that you're rolling down, the one which is very steep. You will reach the floor faster. Alright? Bigger the surface area, the higher the rate of diffusion. This is the same idea as I talked about before, surface area to volume ratio. State of matter. Diffusion is very slow in solid, followed by the liquid, and faster in gases because the particles are most further apart. Let's take a while to breathe first to remember PGEM for the diffusion. The many particles is involved. The gradient we are talking about is the concentration gradient because we are talking about mostly the solute that is dissolved. No energy is required, and it can take place with or without a partially permeable membrane. For osmosis, it will be very, very specific. Only water molecules, pure water molecules will be involved, not water vapor, but water molecules. High water potential to low water potential is a specific gradient that we use for osmosis. No energy is involved, but we definitely would need a partially permeable membrane. The PGEM involved here for the osmosis is a bit different from that of diffusion. That the differences you want to know them clearly. If I word it out, it would be the net movement of water molecules, high to low, down the water potential gradient, down a partially permeable membrane. The format of answering the questions will always be like this. Got to make sure that you follow the what is listed in the PGEM for each of them. Hence, the need for us to define what's potential first. Imagine you have a lift of many people inside, and it's full of people. When you reach the first floor, the first thing you will do is you want to get out of the lift. You have very high water potential, which is a tendency for you to move from inside the lift to outside the lift. Imagine all of you inside the lift are replaced by the water potential that is actually of the water molecules inside a solution. That would be the same way to look at water potential. Tendency of the water potential to move from its original solution to another. The higher the water potential, the highest one being pure water, the more water molecules will want to move out of it. As you can see, I have mentioned and I keep emphasizing the fact that it's water molecules that is moving and not just water. Water indicates the entire solution. If you were to tell me that water is moving, you are saying the entire solution move at the same time, which is wrong. We should be mentioning water molecules are moving instead of water. The types of solution here that we are looking at is, if it's a low water potential solution, the 80% of the sucrose solution, for example, 80%, 20% will be water molecules. Hence, 20% is very low water molecules. Then you will be asking me, then how do I know how many percent of the water molecules is considered high or low? Water potential is always compared to something. That's why we say higher water potential or lower water potential. It is compared to the cell, particularly per se. So when we compare it this way, it should be very easy to tell which one has a higher or lower water potential. For a high water potential, you can tell that the highest is pure water. A 15% sucrose solution means that 85% of it is actually water molecules. Think about it. If your solution is 85% water molecules and you put a cell inside this 85% water molecules, without even thinking very much, you will be able to understand that this solution, 85% water molecules, would be considered high water potential compared to that of a cell. Let's take another 20 to 20 seconds to understand these two terms first and how did I arrive at the percentage for the water molecules. Understanding these two, we are going to talk about what is going to be possible passing through the partially permeable membrane. The partially permeable membrane is a gate, but it doesn't allow everything to pass through. Substances can pass through. As for now, whatever we have learned will be glucose and other one sugar. One sugar, it means simplest form of sugar, which we will learn in topic 3. Of course, water molecules would be able to pass through the partially permeable membrane. Those that cannot are the large ones. I call it the three S's: sucrose, starch, and salts. So what exactly are the partially permeable membrane? The cell membrane would be one of them. If the question mentioned this thing called the Bisking tubing, you will definitely see it very soon. This is an artificial partially permeable membrane. Now, good to go. This slide just shows you how the water potential is involved in the answer and how the phrasing and order is actually done. This is very similar to the one that you've seen when we look at initially the PGEM. I'll leave you to read this on your own, but always take note that you must talk about the difference in the water potential and the net movement of water molecules from where to where across the membrane by the process. In the experience that I've taught so far in many years, most people always forget this. Your story can be very, very long when you involve PGEM completely, but ultimately you forget to write the name of the process that is involved, and that is a serious mistake on its own. For the last part of today's lecture, I'm sorry if it sounds a bit faster because I'm having a runny nose, and we are going to look at how osmosis affects both animal cells and plant cells. Alright, so please bear with me. For the microscopic level, we're going to look at, if I mention the word microscopic, it will be something which you can see. If it's microscopic, it will be something that happens beneath. Dividing it into two will be able to let you see what happens exactly when this animal cell is placed in a solution of higher water potential. When they say higher, it means that the animal cell has a lower water potential. Hence, the water molecules will enter the cell from the solution of higher water potential to the one solution of lower water potential. The solution of lower potential is the cytoplasm by osmosis. Ultimately, the cell will first swell. Important to note that. Number two, then they will finally burst. These two diagrams here shows you very nice diagram of the cell about to burst. In fact, when the cell is burst, you probably only see fragments like this. Burst, nothing left. Alright? How about if you put at the cell into a solution of lower water potential, you'll be good at saying that the animal cell has a higher water potential. First, water molecule leaves the cell from the solution of higher water potential, the cytoplasm, to the solution of lower water potential, which is the solution around it. It exits by osmosis. The cell will shrink first, number one. Number two, it will go into a state called crenate. Why do we say crenate? Because of the spikes that appear. If I were to draw it for you, it will look something like a star, a very distorted shape. All these are the spikes. This cell is in a state of crenation. For the plant cell, the story will change a little bit. If you put a plant cell into a solution of higher water potential, you'll realize that osmosis takes place, but the water molecules enter the cell sap. Because of the large central vacuum, we use the word cell sap, as I mentioned to you before. Number one, the vacuum will increase in size, definitely. The large central vacuum pushes the cytoplasm against the wall, definitely. As this gets larger, the cytoplasm will be pushed against the cell wall. What happens is, the pressure exerted by the water molecules will give you this thing called turgor pressure. The cells swell and eventually become turgid. Turgid is very important for the plants because it helps you to keep the leaves stretch out to receive sunlight, keep the plants upright. We will not dwell so much on this, only when we cover the plant topic, then I will let you know. Just remember, this state of the plant is called turgid. When we look at the other way around, this will be a bit more tricky. When it's in plant cells that we put the plant cell in lower water potential, from the cell sap, the water molecules will exit. And when it happens, you realize that the vacuum strings, the large central vacuum strings, how do you know? There it is. Pull the cytoplasm away. When you pull the cytoplasm away, pay attention that this is actually the cell membrane. Originally, the cell membrane will have touched the cell wall. When you pull it away, as the cytoplasm shrinks away, you will get the cell membrane very, very obvious. Please don't be confused. Alright, so plasmolysis happens. I will not prefer you to use this word, just use the word. The plant is actually plasmolysed. Why do I use this? Because P is for plant and it's easier to remember compared to flaccid. So the plant goes into a state of plasmolysis. The plant is plasmolysed. The plant cell is plasmolysed. Hence the next question, what is all these things here? Remember I said cell wall fully permeable. So if you are in a solution of lower water potential, it can enter the cell wall and it will be located here. It's a common question to ask what is located in between the cell membrane and the cell wall. Please take note that because the cell wall is fully permeable, the solution that the plant cell is immersed in will be located at the spaces there. Finally, to go will be the last process of today, which is the active transport. It's a very new process to you, and the PGEM involved will be these. Please remember, no water molecule must be involved here. Any other particles can use active transport. It is reversal. Instead of high to low, it is low concentration to a high concentration. Energy is released from aerobic respiration. These two must be stated together, please. And you definitely need a partially permeable membrane. Let me summarize it. The net movement of particles where they have going against from low to high. If you are going to climb a mountain, you need energy. It's the same logic here. So to carry out active transport, you need energy. This energy needs to come from aerobic respiration against, because you're going against the gradient, you're going against Mount Everest, you need energy across the partially permeable membrane. The energy is from aerobic respiration. The definitions of each of them are very easy, so I prepared a tutorial for you to actually try. The notes I will give it to you soon, but I actually hope that you will try and summarize the three processes together in a table for summary, and let me mark when I see you in week 5. We will be in time with 3 hours in week 5 to prepare for the WA, including the drawing of one graph. Alright, so need not worry, everything will be fine. Okay?